Hi, everyone. Good evening. We are here tonight for a very special webinar, and we're going to let people wander in for the next minute or so, watching the number of participants crank up. It looks like we had well over 90 people registered for the evening, and so I'm very excited to be able to be here with all of you and share the information about Ovid and Graviton and the new ROCK2 inhibitor that they're developing for CCM. So I want to start with a little bit of housekeeping. First off, we have support group immediately after this, and Darla will be putting the link for the support group into the chat at various times throughout our meeting. All you need to do is click on it, and it will bring you directly into support group at the time that it starts. And then also, at the end of the presentation, there will be time for questions and answers. Please use the Q&A at the bottom of your screen in order to do that. Please don't put the questions in the chat because I will not see them. I won't be looking there for them. All right, I'm going to share my screen now. And I believe we can start from here. So this evening, we are going to be speaking with Dr. Jeremy Levin and Dr. Amy Melsaether about bringing ROCK2 inhibition to cerebral cavernous malformation. We have been working with them since last May when they first announced their partnership and their work in this area. And they have become good partners of ours in the meantime. And I just want to share a little bit about each of them. Dr. Levin is the chairman and CEO of Ovid Therapeutics, which is a public company developing novel medicines to treat epilepsies and seizure-related disorders. Before Ovid, Jeremy served as president and CEO of Teva Pharmaceutical. He was also a member of the executive committee of Bristol Myers Squibb and the global head of strategic alliances at Novartis. Jeremy is currently the chairman of the board at Althea and a member of the board of directors for Bio, for which he's the immediate past chairman. Jeremy earned his bachelor's with honors in zoology, his MA and Doctor of Philosophy in chromatin structure at the University of Oxford, and an MB Sure at the University of Cambridge. Dr. Amy Melsaether serves as the Chief Medical Officer at Graviton, the partner company to Avid in this endeavor. She's bringing with her a depth of clinical and academic experience acquired over nearly 2 decades. Her recent role as Senior Vice President of Clinical Research at Equilibre Biopharmaceuticals reflects a commitment to medical innovation following her tenure in academia. Dr. Melsaether's work as Associate Professor at Mount Sinai and NYU is notable for advancing PET MRI applications in breast cancer. A recognized leader, she achieved fellowship in the Society of Breast Imaging and played a pivotal role in shaping breast imaging curricula. Dr. Melsaether's academic foundation includes a distinguished BS from the University of Wisconsin and an MD from UCLA, augmented by her Howard Hughes Fellowship at the NIH. Her career is marked by a passion for translating complex science into tangible medical progress. And I'm so glad to have both of them here with us today. Today, our webinar structure is going to be that Jeremy will be speaking first about the community-first approach, how Graviton and Ovid are working together to potentially bring a new medicine for CCM. And then Amy will follow that, talking specifically about the science, what it is that is special about this ROCK2 inhibitor, and why this is an innovative approach for treating CCM. And then, as I mentioned, we'll have an opportunity for questions and answers. Okay, Dr. Levin, please take it away.
Wonderful. Hello, everybody. Thank you so much, Connie, for giving us the opportunity to meet the families that are out there. And I hope that today we can give you a glimpse at what we're trying to accomplish. We're very excited by something that is important, and working with you, I think, will make a huge difference. Before I start, please note that as a public company, I'm obliged to ask you to take note of this. This really refers to the forward-looking statements that we might make, but those should be adjudicated with what we actually are doing. So please study this at the later date. It's important. Let me first of all take the opportunity to introduce you to a wonderful company, Graviton BioSciences. Graviton are absolute experts in the field where we work and we will be working. Their focus is really on a very special protein called ROCK2, and the medicines we are developing are those that essentially interdict this, stop it working. They've done this before, actually, so that's what's great about it. The leadership of Graviton's company is Dr. Sam Waxel, who's has devoted his whole life to finding medicines. Indeed, he's probably found more medicines today in the marketplace than anybody else. I think the figure is actually 11. But in this particular case, he's also found a medicine which is called Rezurock. It's in the area of ROCK2 inhibition, not quite the same as ours, but very good medicine as well. So now we're going to replicate it. They are experts and they know how to find these medicines. So we're a biopharmaceutical company. Both companies are located in New York, so we're very close to each other, actually, and we work together very well. One of the things that characterizes Ovid is that we focus on disorders of the brain. Our job here and our commitment is to develop the best possible medicines that we can for the brain. And we've done it, and we continue to do it. Indeed, a little later this year, the first of those medicines which is in phase 3, it's a medicine called soticlestat, designed for rare epilepsies of the brain. Hopefully, we'll get results that show us we've broken open the area. And with a bit of luck, maybe we'll not just stop seizures, but have a chance to start to cure the disorder. Not sure about that, but we're trying. But one of the things that makes us different from others is that we focus on the communities. That's a very important component of what we do. Why do we do that? Well, we know that every time we develop a medicine, we change the lives of those to whom we give it. That's not something done trivially. You want to do that with seriousness, complete commitment to the individual, and complete commitment to the community that supports them. And we've done this repeatedly. Our goal is to build ourselves into trusted partners of the community and for them to understand who we are and for us to understand who they are and what's important. We have a track record of doing this. Indeed, in the area of Angelman's, we were the first and only company ever to get into the area. We developed an endpoint working with the community, and then because we had that endpoint for clinical trials, many other companies started getting into that area. That's a real triumph. And at the end, we provided that endpoint to the community so that they could provide it to the entire area of research into Angelman's. And I hope we've pivoted there. We're going to do the same here. We'd like to. This is who we are and what we experience all the time. This is a partnership in its totality. Now, what's really interesting, as you think about that, we have great skills in developing medicines. This is what we're very good at. It's not about us. Ovid and Graviton are a genuine, true partnership. As we link with the community, is essentially a powerful combination. It's a combination of community-driven initiatives, a deep, deep understanding of how to inhibit that protein ROCK2, and then more importantly, as we start to do that, is to understand how to get the right molecule at the right time. And the molecule we're working on at the moment is a special one called OV101. It's an inhibitor, which means it stops the functioning of that protein. And Amy will tell you much more about how that works shortly. And it emerges, and we believe it will be fundamental, and not just emerging, but it will change the direction, if it's successful, as one of the great opportunities to change CCM's root causes and to address it. So we're very excited by this. And as we develop this partnership, we bring different things to the table. Their deep interest in the science, their skill, their experience in developing the molecule, and our capability of not just working with the communities, but also working with the FDA, working with the others outside of this, and developing a medicine. So it's a partnership which is strong, but it'll be made much stronger by working with you. And we're looking forward to that. And just to give you a glimpse of who this team is, I won't go through them, but Sam Waxel, Dr. Sam Waxel, a many-decade colleague and friend of mine, somebody I respect deeply, is in the top left. He's done it before, and we're going to do it again. Amy, as you can see, is there. But take note of something else. Many people that you'll meet along the way, Raquel Cabot, Patrick Samir, Juliet Sai, Manoj Malhotra, Ray Wu, Pushpam Bhattarathi, and indeed Alexander. All of us are here. We come from different backgrounds, but take a look at those backgrounds. They say something. We have a lot of experience focused on solving for this problem. And from our perspective, it's one which is a nearly unique combination to be seen in the industry. Two relatively small companies coming together to solve a very important medical issue. But it's not just about before. We are incredibly grateful to be working with Connie, both as an advisor and guide. She sheds light on so many different things, as you all know. She's built not just a tremendous organization, but also the way of understanding how those from the outside can and should work with that organization. And we're delighted to have that opportunity. So we're most grateful to her. And if I could clap and everybody could hear it, you'd hear me clapping for Connie right now. I think also it's very important to understand we're very medically focused. And in doing that, one of the things that we have focused on, is a really important contributor to this, and that's Dr. Awad. We are very grateful to have the opportunity to work with him. And from our perspective, this is a way that we get better at what we are doing, understanding the medicine, and we can actually then have somebody working with us side by side as we march down into the clinics and understand the impact we're going to have. So how do we work together? Well, that's pretty straightforward. We start with you. Truly, that's how it starts. We start with you, the community. For us, that's just everything for us. And if you think it's a beginning of a journey, we'll be by your side and you'll be by ours for years to come. And we hope as we do that you'll join us, you'll come visit us, we'll see you. We've loved getting to know the community, actually. We've met many of you in the last year in the meetings in Miami, the Nelson family, David Borland, Darla, and several others. We've spoken and heard from remarkable people. Rachel Paverman, gosh, what an extraordinary person. Frankly, all of us should be reading her story, and we will do, since she was diagnosed with CCM. And, really what she's done with that, it's tremendous. And we've learned so much from you. So thank you. This is a beginning. Thank you especially to Linda and the CCM1 Hispanic mutation community. Trent's poster, by the way, posted last year in the scientific meeting, really instructive. Oh, by the way, I think there's a birthday there, if I'm not mistaken, so perhaps I'm right. Happy birthday. You have accomplished a huge amount, a huge amount. So let's get on with this. Let's dig deep. I've got Amy right with me, and I'm going to hand over to her and enjoy this because I'm going to enjoy it. It's a very special occasion to hear from a very special person.
Hello. So thank you very much for being with us tonight. Thank you, Connie, for hosting this webinar. And thanks so much, Jeremy, for your kind introduction and for your background on Ovid and the Ovid Graviton partnership. I'm going to give, as we talked about, I'm going to give a little bit more detail on ROCK2 inhibition and why ROCK2 inhibition may provide a multifaceted approach to CCM. So we're really excited to bring this medicine forward for this specific disease, in part because there are many mechanisms through which ROCK2 can work to hopefully have an improvement for people with CCM. So this is just a review. I think many of you are going to be familiar with these images and with CCM on the whole, but just to remind everyone here on the left, this is an MRI, a T2 MRI image of the brain, and what we can see in the upper right corner of this brain is a lesion that doesn't belong, right? So this is a tangle of vessels, and around this tangle of vessels, we can see what black halo, and that black halo is actually iron or some blood deposition around the lesion. We can see in the middle image here, this is a different kind of image. It's a susceptibility weighted image. But what this is showing here, it's highlighting areas in the brain where there are concentrations of blood. And so what we see here is that there are many innumerable areas throughout this person's brain that are affected by blood. And this is a representative image of a person with familial CCM and multiple lesions. And then if we move over to the far right, what we see here is a cartoon or a representation of that mulberry-like lesion of CCM. So what's happening here in CCM lesions, the endothelial cells, which are the cells that make up the blood vessel walls, those cells aren't forming their normal tight junctions with each other. So there's a weakened junction. And if you can think of a balloon or any kind of object where you have a thinned surface and then you try to pump water or pump some fluid through it, you'll get an outpouching there. You can be familiar with that, right? And so what's happening in these lesions is you're having your endothelial cells that aren't quite joined as tightly as they should be. You put the blood through that lesion and then you can get an outpouching or a thinned bubble. And that part is vulnerable to bleed or rupture. And that's what can cause the problems in CCM. And so let me just get to the right advance over here. So this is cartoon images of CCM lesions up close. So in the top right corner, we have healthy vasculature. So what does a normal arrangement of endothelial cells look like? And you can see here, they make a ring, and inside that ring, the blood flows. And what's very important about this ring is that there are tight junctions between these cells. And those tight junctions do an excellent job of keeping the red blood cells inside the blood vessel and keeping other cells outside of the blood vessels. And those other cells can be inflammatory cells. In normal healthy vasculature, you won't see inflammatory cells surrounding the blood vessel, but in CCM vasculature, those tight junctions are not reliably able to form. And when you have impaired or loss of tight junctions, then those red blood cells aren't kept as tightly within that ring, and they can leak outside. And when you have red blood cells leaking where they don't belong, you'll notice there's a new kind of cell here. This is a microglia. It's an inflammatory cell in the brain, but there are other cells we're familiar with too, like macrophages and lymphocytes, and those can infiltrate actually in through these impaired tight junctions. And so not only do you have blood leaking out, but you get an inflammatory reaction where you have a recruitment of inflammatory cells. And because of that loss of tight junction, the inflammatory cells can then make their way into the lesion and cause their own complications there, as well as outside of the lesion, where inflammation can cause a number of symptoms, including seizures and headaches. So what happens when we have this loss of the tight junction is we have increased vascular permeability, which means that those vessels are permeable. It means you can get leakage. Those red blood cells can come out of the vessels. You have an increased likelihood of a frank bleed, and then you have increase in inflammation, and inflammation we know can lead to scarring or fibrosis. So these are all things that can happen because you have a loss of that tight junction. Moving ahead. So how can we manage CCM? So what are the concerns with CCM? So once a lesion has bled, there's a very real risk of another bleed. So we know that there are many lesions that may not bleed during a person's lifetime, but when there is a lesion that has bled, that lesion is at risk for another bleed. symptoms from that initial bleed may persist, and even when they don't, the risk of a bleed may impact physical and mental health, and it can influence career and family planning. So I was fortunate to be able to attend the New Mexico symposium on CCM this April. And there was a person who led off, and he told a compelling story. I don't know that I'll get all the details right, but what I wanted to highlight is that this person made a change in their career planning based on their CCM. So this person was a firefighter, and they'd had their first bleed or their first onset of symptoms while they were on a call. And that's very concerning, right? They're in the middle of a call, something's happening, and say, oh my goodness. They went to the hospital, they found out they had CCM and they had had a lesion that had bled. So they're back on the job a little bit later and they have a rebleed and they have those symptoms again. And then this person made the decision based on these symptoms and the unpredictability of the bleeding that maybe this isn't the right career. Maybe it's not the right path to be leading a team who are necessary in emergency situations when their health is not reliable. I think the outcome was the person had a stable period for the next 14 years, but still they had made a big decision about their career planning. And we had a chance at last year's CCM meeting hosted by Connie to hear stories from a lot of people and their families, and other things came up like family planning. And how do people manage birth control in this setting? There are a lot of concerns that come up where we see that symptoms aren't the only thing that affects people's lives. There's also that risk of bleed. So what if we could address symptoms and that risk of bleed? How would that look for patients? How would that look for their lives and their families? And so what we want to talk about here is ROCK2 elevation. What does that mean, and what might it mean to get rid of that ROCK2 elevation? So in CCM, there are 3 proteins, CCM1, 2, and 3, and they come together to form a complex. And that complex somehow works to limit the activation of Rho A. So it blocks or inhibits Rho A. But when you have faulty CCM proteins, that complex can't form, and Rho A levels go up. Now, Rho A is right next to ROCK2. Its job is to activate a number of downstream proteins, and major proteins that it activates are ROCK1 and 2. So here we have on the left, you can see that cells with reduced CCM1, 2, or 3 proteins are also cells that have increased Rho A. And the science to document this is down in the lower left corner. I don't expect you to be able to read this, but we'll walk through it together. So what we can see here, this top line here is Rho A levels, and the far left WT means wild type. That's a fancy word for normal. So in normal cells, you don't see Rho A. And here we have cells that don't have CCM1, CCM2, or CCM3. And what we can see is when you're missing any of those proteins, you have very elevated levels of Rho A. And they quantified, this is work by Borikova et al. They quantified these levels and found that in cells that don't have CCM1, 2, or 3, you have 20 to 35-fold elevations in Rho A protein. So why is that a problem? What does Rho A do? So Rho A lives near the cell surface or the cell membrane, and so it can receive a lot of incoming signals. So it can receive stress signals. For example, maybe you have hypoglycemia or low levels of glucose. Maybe you have some inflammatory signals. You have TNF alpha. Those signals activate Rho A. Rho A takes in those signals and then activates downstream other proteins. It passes on that message. And one of the proteins it passes the message onto is ROCK2. And ROCK2 does a great job of broadcasting that stress signal to a number of downstream pathways. And what we can see is that after ROCK2 is activated, it activates a number of processes. And these processes lead to increased cytokines and inflammation. They lead to fibrosis. They lead to vascular leakiness, and they can lead to vascular damage. And we'll move on to the next slide here. So to summarize ROCK2 and CCM, why does CCM have increased levels of ROCK2? Well, mutations in any of the CCM proteins, again, impair formation of that complex. That complex normally inhibits Rho A activation, but when that complex doesn't form, Rho A goes up, Rho A directly activates the ROCks, and then ROCK broadcasts those signals. ROCK2 signaling can be thought of as stress signaling. And things that happen when cells are stressed are that they stop their outward growth. They conserve their resources, stop their outward growth. They're not growing towards each other. They're not able to form those cell junctions. You also get increased inflammation and increased fibrosis. So what might happen if we could really block ROCK2? So what we hope we can do when we block ROCK2 is to mitigate stress signaling or to stop or decrease stress signaling. So we hope that stress signaling can be halted and the cell would then be free to return to its balanced state or to homeostasis. So on the left here, we have another picture, cartoon, or diagram of what happens when you have too much or hyperactivated ROCK2. And these are endothelial cells, and you can see here that there's a weakened junction. There's space between these cells. When there's space between the cells, you have the red blood cells coming out, and again, you can have inflammatory cells coming in. These green lines throughout are stress fibers. Stress fibers are something we won't talk too much about, but they're also related to ROCK2 activity, and you get a number of disorganized stress fibers when you have too much ROCK2. When that ROCK2 is inhibited, we see the stress fibers are aligned in a normal, orderly fashion, the tight junction is restored, and the red blood cells are able to stay within their normal blood vessel walls. So what about our ROCK2 inhibitor? So the OV888 GV101 capsule is a selective ROCK2 inhibitor. It's actually, it's a molecule we're very excited about. It has very, very high selectivity for ROCK2 over ROCK1. This is new. As Jeremy talked about earlier, our team developed Rezurock, which was a ROCK2 inhibitor with much less selectivity. And we are really thrilled that we have this really best we've seen selectivity for ROCK2 as compared with ROCK1. And why is that important? Well, because ROCK1 and ROCK2 are very close, but they are different. And they have different functions within the cell. But because they are so close in how they look, it's difficult to design a drug that inhibits one and not the other. So we are very fortunate to have a drug that inhibits ROCK2 much more than ROCK1. That's especially important because, as we've shown in our in-house labs, and I'll show you some of our in-house lab work in a minute, blocking ROCK2 actually can limit the effects you get, the therapeutic effects you get by blocking ROCK2. So when you block ROCK1, you don't see the same anti-inflammatory effect that you see when you block only ROCK2. Other things to think about are, in some cases, blocking ROCK1 can cause some side effects that ROCK2 doesn't cause, and so it can limit the clinical utility of a drug. So what happens if we block ROCK2? We're back to Borichova's work from 2010. And what we see here is that there's potential restoration of vascular structure by blocking ROCK2. So this is another difficult image to work through, but we'll work through it together. So you can see on the top line here, these are normal endothelial cells. And they're put here in a plate. And then we photograph the plate, or the plate was photographed at several time points, several hours out. And what you can see is that normal endothelial cells will come together around 3 hours and start to form these tube-like, vessel-like structures. Now, this experiment was done for cells that were lacking CCM1, CCM2, and CCM3. This is just a representative image from CCM1, but the story is the same for all of them. With no treatment, cells that have these mutations don't come together to form these normal vessel or tube-like structures. And then they did treatment with a couple of different treatments. So one was a drug that blocks ROCK1 and ROCK2, although blocks ROCK2 a little bit more than ROCK1. And what we can see is that over time, some of that ability to form these tube-like structures is restored. But when selective ROCK2 is blocked through an RNA mechanism, which means they just block all of that ROCK2 protein formation, we end up with vessel tube-like structures on a timeline that's very similar to normal cells. So what we're taking from this image here is that when ROCK2 is blocked in people that have, or in cells that have lack CCM1 proteins, you can restore or you can compensate for that lack of the protein. We'll go on to the next slide here. The other things that can happen when we block ROCK2, these are experiments we've carried out in-house in our own laboratory with people very experienced at working on ROCK2 inhibition. And we can see two things happen. And actually, these are a little out of order. We didn't get the slide quite how we wanted it to be. But the second thing that happens is a potential restoration of immune balance. So what we're seeing is that when you treat cells in an inflammatory environment, but you treat them also with our GV101 or OV888, we see decreased expression of these molecules that typically cause inflammation. So here you can see in this picture here, the levels of pro-inflammatory molecules are decreased, and they're decreased in a dose-dependent manner. That's important. That means as you give more drug, we see less and less of these inflammatory markers. And conversely, and this is unique to ROCK2 inhibition, we see increases in markers that cause tolerance or wrap up immune response or act to stop inflammation. So not only do they decrease markers that cause inflammation, they also increase markers that can ameliorate or end or calm an inflammatory response. And we see that here on the far right. Now, when you have a decrease in inflammation, you can also have a decrease in fibrosis, and that's what we're seeing here. So when cells are treated with a molecule called TGF-beta, they go into a fibrotic state. When we treat them also with our molecule GV101, and these are again complicated figures to read, and they're actually reversed from these down here. But what you can see is that again, there's a dose-dependent decrease. The higher levels of the drug are on the left here. So there's a dose-dependent decrease in expression of several profibrotic molecules. In this case, we've singled out alpha smooth muscle actin, as a single type of collagen here and connective tissue growth factor, but these are representative of a broader pool of fibrous markers that are all decreased when cells are treated with GV101 OV888. And so this is actually, how does this bear out in an animal situation? So this is work by McCarricher et al. In 2020. And what they did is they looked at mice with impaired CCM. So in this case, it's mice with CCM3 mutations. And what we see here is the importance of ROCK1 versus ROCK2. So then when you have decreased amounts of ROCK1, we really don't see much of a change in the percentage of mice who have CCM lesions. And we really don't see a decreased volume of lesions or decreased amount of blood in these lesions. But when you have mice with CCM mutation and also lower levels of ROCK2, you see that the number of mice or the percentage of mice with lesions decreases. And you can see also that the blood or the bleeding of these lesions significantly decreases in the treated or the patients with normal levels versus decreased levels of ROCK2. So to summarize, what happens when we block ROCK2 in mice with CCM mutations is that you see fewer CCMs in these mice. So to summarize, when you're blocking ROCK2 and CCM, what can you do? Our hope is that you restore that tight junction, improve the vessel tube formation, decrease the release of inflammatory molecules, and interrupt that fibrotic or scarring reaction. All of this acting to restore balance in the brain. So one question I thought we'd address for the community, something that comes up often is, how does blocking ROCK2 relate to other treatments that have been tried, other treatments that might be available? And so three that come to mind are atorvastatin. So atorvastatin is a statin drug, and I think many of us are familiar with statin drugs. So they are designed to inhibit cholesterol synthesis. One of their side effects or other effects, drugs often have multiple effects, is to attenuate ROCK signaling. So they decrease ROCK activation also. But in order to get that ROCK activity, you need very high doses of statin. And we'll see. There's an ongoing trial that's just been recruited, and we're very excited to see how that results. Propranolol is another agent that's been tried, I'm sure as you know, in CCM. That acts by beta blockade, and we don't know exactly how it acts in CCM. What we do know is that it worked on infantile hemangiomas, which are a lesion that has some similarity to CCM. And there are some case reports where propranolol has worked for CCM, but in larger trials, it hasn't been borne out yet. And then, of course, there's a REC-994 trial. They have a drug that acts by reducing superoxide, which is a reactive oxygen species, which is a mediator also of inflammation. This is a great mechanism to reduce inflammation. And reducing superoxide may be one of the things that ROCK2 inhibition can do as well. And so where are we in progress with GV101 and OV888 capsules? So we are testing in healthy volunteers, and we're thrilled to let you know we're going ahead testing at in healthy volunteers at multiple doses, and our participant dosing has completed. So we finished what's called multiple ascending dose study. The patients have all been dosed, the safety data is in, but it's still blinded, so we don't yet know the exact results. We're happy to share them when they are available. We know already that there were no serious adverse events, and we're excited to share that safety profile with you. We think it will be before the end of June, so we have that to look forward to. And I just wanted to end with this. Together, the community, Ovid and Graviton can make a difference. We're aiming to restore balance in CCM with ROCK2 inhibition and to bring innovative new treatments to this community. So thank you again so much for being here, and we're excited to answer any questions you might have, and excited also to see you in Toronto. For those of you who are going, we will be there in mass.
Thank you. Thanks. And we have questions coming in our Q&A, and thank you everyone for using the Q&A. Please go ahead and put your questions there. You both did a wonderful job. Thanks. We have our first question is really about whether or not this medicine seems like it could be something that could help people with the sporadic form of the disease. We've had multiple people ask that question. And either one of you.
Amy, why don't you grab that?
No problem. So we are hoping very much that this is something who can help people with sporadic forms of the disease, right? So what we want to really look at is in people that have a lesion that's declared itself, that have a symptomatic lesion, we know the treatment available is surgery, right? Or watchful waiting. And that's what you have. And surgery, neurosurgery can be a wonderful thing, and it can also be a scary thing, and it can also carry risks of its own. And what we'd really like to do is stabilize those cell-to-cell junctions and decrease the inflammation. If we can stabilize those cell-to-cell junctions, we really think that we'll be able to decrease the risk of a bleed. So what we're investigating is in people that have had a bleed, can we decrease that risk of another bleed, which I think is the real question. If we can stabilize that, then our hope is people can go on without anxiety, without considering, am I near a hospital? What do I do? What do I do for my planning? So what we're really hoping is to make it a non-issue, to say we had this once, and we really hope we can provide this treatment, and we hope that treatment will be able to prevent a future event.
What effect does ROCK inhibition have on the other cells in the body, or is it targeted to only the CCM cells?
I'll start, and then Amy, you'll go after me if that's okay.
Perfect.
So ROCK is a really interesting protein. It really is part of a complex within the cell, and it becomes overactive and raised when disease strikes that cell. So the whole principle of this is to knock it down when it's up. So effectively, in a normal cell, it won't have an effect. It will have an effect in a cell that's showing abnormalities. And its ramifications, once it's knocked down, are broad-based in that cell. And as Amy mentioned too, in the case of ROCK2 inhibition, what happens is that you then reform these incredibly interesting actin filaments. And in fact, your microtubules in your cell, which act a little bit like tent poles, start to straighten out. And what happens is that the cell starts to readjust and get its shape again. Now, in a normal cell, that won't happen because they're already normal. And in addition to that, you have something else, the cell junctions, which are also disrupted. By knocking down the ROCK, you bring them back together again. Again, in a normal configuration, that's not necessary and doesn't happen. Amy, do you want to expand on that?
I think that's exactly right. So we can think of ROCK2 as a stress mediator. And so in normal cells, we're not seeing active ROCK2. And so like Jeremy said, we don't expect, or we haven't seen in all of our work on ROCK2 that we have a negative effect on healthy cells, right? Because ROCK2 really is a mediator of stress, fibrosis, inflammation.
So, is this a medicine that you would take for a lifetime, or do you just take it until the lesion is stable, or how would that work?
Well, I think there are two ways of looking at this. First of all, we will learn a lot in the clinical trials exactly how this works, okay, and how much it affects what are the lesions themselves. And that's a good thing, because what you do then is then you can decide how long somebody has to take them. But my assumption is that this is a little bit like metformin for diabetes, propranolol for heart or hypertension, or something like that, or even statins for cholesterol. You take them, you don't notice them. It keeps the actual lesions away, and that's what would then do for the rest of life. I myself, for example, take a statin, don't even notice it, and will probably have to take it for the rest of my life unless somebody comes up with a magic wand that converts my high cholesterol to nothing.
Okay. Do you have a timeline for the trial? Folks are actually asking in the Q&A, so when is this going to be available and approved for everybody? But what is your timeline at this point? How far out can you see?
Hey, do you want to catch that?
Right, so we are, like I said, we're working to complete our phase 1, which is our safety data in healthy volunteers, and this is a very important step. What we're seeing now is good safety data at this point, and we'll be able to release that hopefully in the coming month. And then we're working very hard to develop our what's called a phase 2 or trial of this medicine in patients with CCM, and we're working together to finalize those plans. So we're hoping to be able to open the study. The goal is by the end of this year, and we'll see how we come along with that.
And then they're also asking about clinical trial sites. Do you have a sense of how far and wide the clinical trial sites will be?
I think you need to think about this as our goal is to affect this around the world. So as we've done with other trials, we'll pick the best sites wherever we can around the world and start there, and then in fact move very quickly throughout the different nations one by one. So we haven't yet made final decisions on this, but we most certainly will do that. Amy?
I think that's exactly right. The goal is to make this as broadly available and to be as inclusive as we can be.
Do you expect the medicine to be a pill or a more complex intervention?
You know, it's such an interesting question. Sometimes simple is really good. I started medicine and thought much about that. And for me, the most important thing is knowing that if you take a pill, you are going to change somebody's life. And so I prefer to take a pill where it's possible. But what's interesting about this particular molecule, and that's our intent, by the way, we have gel caps at the moment, and I'm sure we're going to have others, much more interesting ones down the line. That's the nature of it. You start really with a solution, you make a gel cap, and then from there you move on to the final pill. It's a process which I've done, and I know that Dr. Waxel has done many times. But at the end of the day, what's really interesting is that this particular molecule is amenable not just to pills, but also to infusions. But we don't expect to be doing infusions on this at this stage. It's overwhelmingly for this type of chronic use, you'd want to have a pill. Amy?
Exactly. So we're fortunate that we have a number of different options. But for CCM, we're looking at right now it's a capsule formulation, which is an oral once daily dose.
Okay. And the same person added, I'm also interested in that magic wand for high cholesterol.
So are we. So are we. That'll be the next project. What do you think?
Okay. Uh, so again, a lot of people asking about trial sites, New Mexico, Australia, et cetera, et cetera. So would being on a different trial drug make me ineligible for this trial? And what they might be referring to is not necessarily the clinical trial for REC 994, but atorvastatin, propranolol, other drugs that people are using for CCM.
Good question.
That's a really good question.
Let's define exactly what the entry criteria is, and then we'll tell people. In general, this is a decision that the individual and their doctor must make. It's very important. We want to be sure that we are seen not just running our trial just because the person has something that we're interested in, but also we look at each individual as a whole individual. What's best for them right now, and what would give us the most uniform a mix of people in a trial? Because what you want to do is you want to be able to be sure, one, that it's safe, and you don't want to have any opportunity for it to be unsafe, and we've got no reason to believe it isn't safe, but you want to reduce the number of variables. And one of those variables is having medicine. So a robust discussion will be held with each of the physicians. I think that's the way I would look at it. How would you look at it, Amy?
Right. So one thing we really want to make sure is that any effect in a clinical trial can be attributed to the drug that we're testing, right? And so we're definitely aware that a lot of people have been participating in trials for statins and for propranolol. And that's a question that we're working on right now is, is it feasible to keep people on these medications? Can we still test the outcome of our drug? And that's a decision we haven't made yet. So we'll let you know, as Jeremy said, when we get the finalities of the trial decided. We'll go from there.
And going back again to trial, will the trial accept all kinds of volunteers? For example, someone in their 60s with multiple CCM1 lesions and seizures. And how can they enroll? We're already
We, as a rule, we try to be as broadly inclusive as we can while maintaining safety for our patients. We haven't decided yet on the final inclusion criteria, and we will absolutely let you guys know. But our basic aim is, we think we've got something good. We're excited to bring it forward. And we're grateful for people who want to participate and join with us. And as you know, I think Jeremy's really made a good point. Our goal is to be as inclusive as we can. And so we work to find ways to broaden those inclusion criteria.
Can you share a little bit more about the safety trial, just in terms of how long were people in it, how many people were in it, just the composition of what a safety trial is?
Amy,
Absolutely.
you were doing that. Why don't you go ahead, please?
I'm very close to this one, so absolutely, I can give some background here. So, so this particular molecule, we have tested it in multiple ascending dose trial and a single ascending dose trial before that. In the multiple ascending dose trial, it's a randomized placebo-controlled trial, and it's was 8 people in the first dose group and then 10 people in each subsequent dose group. The doses ranged from 100 all the way up to 600 milligrams. Um, and so 2 people in each group will get placebo, and 6 or 8 people in each group will get the study drug. And then we watch them. They get that drug daily for 7 days. They're kept in-house, and we monitor all sorts of things. We monitor EKGs, we monitor all kinds of different lab tests, we monitor physical exams, neurological exams. So we are very grateful to our volunteers for these studies who sign up for a lot of evaluations, and those evaluations really help us get a sense of what we would need to look out for, or when we would need to do testing, or even how we can push testing out a little bit and make the trial a little bit easier for people who come after them. So that's the composition so far. It's been 1, 2, 3, 4 dose groups. And then after that, a group we actually studied for food effect. So another 12 people were dosed with this drug. Those are only dosed 1 day and then another day, but the rest of the people were dosed for 7 days in total. And then as another background, there's a lot of work that goes even before you're in humans. And so we've done a lot of that background work to test the safety there too.
Jeremy, did you want to add anything?
No, I mean, one of the things that everybody should be reassured in is that our goal in any clinical trial is that you should, as an individual, the person running that trial, be prepared to take that drug and give it to your children. And that's the way I've looked at every trial I've ever run. Safety first, make jolly sure of it. And Amy's done a phenomenal job in reassuring it and putting together exactly what you have to do to assure that it's a safe drug.
All right, my next question is about post-approval education.
Excellent.
If the drug is eventually approved for commercial use, how will you educate the neurological community, and more importantly, PCPs who rarely understand CCM and are hesitant to understand it and thus hesitant to prescribe for it?
What a wonderful question. You know, it's actually not alone. In the case of CCM, launching a new drug, and I've done quite a number in my life, is a very subtle and thoughtful process. Now, it depends critically on something which is unusually real. In this case, Connie, it's you. You're one of the core areas of education that one starts with. One starts with the community. And often the community knows more about this because it's their participants than actually the physicians. But it's then the job of the companies to mount a campaign with medically trained individuals to talk to those who are who are caring for the families as physicians. And it's quite sophisticated because it's changed fundamentally. It's not just somebody going knocking on a door and saying, hey, let me tell you about this and give you some cookies in the meantime. That's what they used to be as a salesperson. And that's when I started in my business years ago. That's what they did. But now it's much more sophisticated. You do lectures, you write papers, you go to congresses, and you make it something that they should know about. One of the great things about this is if you show great results, physicians and primary care physicians become incredibly interested because suddenly you've got something to offer. And life is fairly important for them. They want to know that they're part of the community as well. So that's the other side to what one does. It's not just telling them about the medicine. You tell them how they contribute to the community. So it will be a very sophisticated plan. It's one has worked extremely well elsewhere, but it's based around medical information because the families are sophisticated enough to understand it, and they should.
I agree.
By the way, I would absolutely love the opportunity to launch the drug. You have no idea. This will give me such great pleasure.
And can we just say that Connie's done so much work on having this disease recognized as an entity, right? We heard the news that the CCM will have its own ICD, I'm going to get this wrong, 10 or 11, 10?
11.
11 code. And when a disease has a code, all of a sudden it's much more treatable. And there are more options. People can name it, people can code it, and people can do something about it. So I can't overstate how instrumental Connie and this alliance have been in really bringing the possibility of medicine to this community. I think that's huge. And so congratulations to the community for all the work they're doing, and to Connie in particular for moving this forward.
Actually, Cody, I just tie you in on Raquel. Actually, for the first time ever, it was Raquel who got the ICD-10 code for Angelman's. Never before. So we've been through this. We know exactly the heavy lift that you've done. So hard and so important.
Yes, it is. And hopefully ICD-11 will be used in the US at some point in the not too distant future. It is used elsewhere now. It's been in use since 2022 in most places in the world. Couple more questions. This is parallel questions. If we have increased inflammation in the brain due to CCM, and then the other person or the other question was if we have dysfunctional endothelial cells in the brain, are they also problematic in the rest of the body if you have the familial form? Either do we have inflammation everywhere in our bodies, or do we have abnormal endothelial cells?
It's a very sophisticated question. In general, if the inflammation has caused the ROCK2 to increase, you will be able to ameliorate it wherever it is, whether it be in the retina, whether it be in the spinal cord, wherever it is. So I think the whole concept of inflammation in the brain is incredibly important, and ROCK2 plays a fundamental role in that. Suppressing and knocking down the ROCK2 activity will have an effect everywhere in the body, for sure, if the ROCK2 is raised.
Amy, did you want to add anything?
Yeah, we can say that because of the CCM complex is active in the endothelial cells in particular, we would expect that this is the place where you're seeing the ROCK2 activation in people with CCM. We wouldn't expect that their whole bodies are in a complete inflamed state. It's localized. But that's not to say, in people that have areas of inflammation, you can see markers of inflammation circulating, but that doesn't mean all of your organs are. I just say, please don't go home and think everything is wrong. That's not the case. It's a local, it's because the proteins are local, it's a local effect.
It's very circumscribed. That's why you see on the CAT scans that you saw in that presentation how localized it is. It's remarkable. And you do find similar types of complexes and in the retina and also in the spinal cord. But we're going to focus first on those with it in the central nervous system.
Is a question, are there any individuals out there that have genetic knockout of ROCK2 as a result of mutation? So in other words, instead of having a CCM1 mutation, is there such a thing as a person with a ROCK2 loss of function mutation or gain of function mutation, I guess it would be?
Super question. Don't have the answer.
I feel like this is something I should know, and I don't. So I will be looking this up tonight.
Boy, I just knowing the biology of ROCK2 as it is, that'd be a tough road for them.
It's tricky, right? I know that. Yeah, I don't know.
Okay. All right. Maybe.
But good question. You stumped us.
Some sort of somatic mutation like the PIK3CAs.
Maybe we just didn't get our PhD in ROCK2, right? That's it. We're in trouble.
I got to go back.
Gotta go back straight away. But it's a good question. Thank you for
one little story that I want to make sure I share before we have to stop. So I was able to visit the Avid offices a number of months ago. And one of the things that struck me is that each of the offices that a staff member works at has a picture of a patient and the patient's story for one of the medicines that they're working on developing. And right now, Ovid is asking our patients if they would like to be part of the CCM wall that they're planning. So if this is of interest to you, the reason that this wall exists is so that the folks who are working on this medicine can see the faces of the people that they hope to help. And if you would like to be one of those faces, then please let Darla know, darla@alliancetocure.org, and she can be the person who helps to get your photo with a photo release, of course, over to the Avid offices so that they can see you. Darla just put her email address in the chat too. Trying to see if there's any last questions. Will the recording of this presentation be available? Yes, it will be. It'll be up on our YouTube channel in the near future, probably in about a week, because we clean up the closed captioning. What questions have we forgotten to ask you on? What other questions should we be talking about?
I don't know. Come on. I think this is
Fair enough. I think we're good. I mean, we had the opportunity to put together what we're looking to present. We're hoping when we have some more information and some final plans to be able to present those as well. We're excited to continue to engage. It's been a real pleasure to be able to meet people who've been so committed. There's a very strong network already, so we're thrilled to be becoming part of that. Thanks for welcoming us in. And
I know what.
I told you most of what we can think you'd want to know.
Come and see Hudson Yards from our terraces. You're welcome. We'd love to meet you. We will get to know you. As I said to you, this is a journey that will take years. Hopefully it'll go quickly, but I can't promise that. It'll go exactly in the right way, so we're medically 100% sure that what we've got is something that will help. But Connie's been there. She knows it. It's not bad view, right?
It's a great view. It's a great view. Go out on the terrace. You look one way and look the other way. I promise you'll be struck. Yes. And also, one of the things that I need to make sure everyone knows, I see still a lot of questions here. Most of them right now are about things that are in the future that we can't necessarily know at this point, like where the trial sites are going to be, or how do you enroll, or when is it starting. We'll have that information for you as soon as we can. And when it comes out, it'll be shared with me. It will be shared with the community. The Avid and Graviton folks will be coming back for another webinar as soon as they are able to share that. So just hang in there. Today was our introduction. It was our opportunity to get to meet each other, and this is an ongoing story. So I want to thank both of you for your time and for your expertise and for your dedication to our patients. I think we are looking forward to having a very long and hopefully very positive outcome with our relationship over the years. So good night, everyone. Again, there will be a recording up in about a week. There is support group. Please click the link in the chat, and Darla will have a chance to talk with you then. So thanks, everyone.
One more thank you to Raquel Cabo at Ovid for
Yes.
working so hard to put these slides together and get us all here together. So thank you for that.
Connie and thank you so much for having us with you. And Amy, what a pleasure. Fun. I work with you, but I don't get to see you like this.
Fantastic to see you after hours. We'll see you again soon. All right, take care. Thank you very much, Connie.
Bye-bye. Good night.